And I'm taping there. Okay, so on page 2435, candidates should know inheritance. So Mendel, and the terms allele, locus, gene pool. You know what the gene pool is? So we are all Homo sapiens, and we have this gene pool for all the variation, that 1% difference between all of us. Okay, that's our gene pool. Well, it's all the genes actually, plus the one, including the 1%. Then when you do crosses, all those terms, gametes, phenotype, filial, parental generations, dominant, recessive, heterozygous, hybrid, homozygous, genotype, phenotype. How monohybrid crosses occur. Get the ratios. No partial dominance. So not that pink, red cross with white gives you pink. And not the, what's the other one called? Co-dominance, where white with brown gives you mottled white and brown. That's crossed out of the textbook. Then XY linkage. All right, and then now, all right. Yesterday we did that. Continuous characteristics. Continuous characteristics tend to be polygenic. So you don't just have people that are short like me and tall like someone else. You've got that range, that bell curve. Okay, that's continuous variation, and that's called by polygenes. A, B, B, C, C, for example. Yes. Just understand what it means. So they might give you a case study and say something about polygeny. I actually now, standing, I can't remember where they did ask it. It's like little things. Okay. All right. So like I showed you yesterday, the more dominant genes there are, the more, the darker the color of the skin. Or the more recessive genes, the lighter the color. Or tallness, shortness. Okay. So that's non-Mendelian because Mendel said tall and short. All right, but now we know that height and weight and, what else? Height, weight, length of nose, everything. There's this whole bell curve range. Okay, it's not just one gene, it's a number of genes controlling those characteristics. Polygenic, so therefore the continuous variation from short to lots of medium height to very tall people, like over 2 meters. Okay, all right, my granny, I could do that, my mom's mom. Could fit under my arm. She was very short. Okay. All right. So now, number 5, how gene mutation may lead to speciation. So when we think of the word mutation, what do we think? Come on, be honest.
Something different.
Something different. I was expecting something else. What else do we think?
Wrong.
Something wrong. Okay. Mutations are natural. We're actually all born with about 6 of them, I've read somewhere. So in other words, your DNA is a little bit different to what it was in your parents, whatever gene it is that you inherited. And a lot of them are neutral. So eye color. Well, there's others, obviously, that you're not maybe seeing in the working of your body. All right, and sometimes a slight change in the DNA can still produce the same protein. So you might have a slight change in the gene for making a hormone, but that one nucleotide difference doesn't make a difference in what the protein is made up of. So, okay, so you can have neutral mutations, and then we all think, oh, mutation, harmful, that green thing walking down the road chasing someone. Okay, and then beneficial mutations. All right, so we're doing number 5, harmful, beneficial, neutral mutations, what these mutations are and how they are caused, and what the impact is. So that's what we're doing now. 4, 6, Vion's not here. Okay, so I will upload the videos onto Google. I've just found the 2. Can I show you the 2 videos? And then the PowerPoint, which has got a little bit of animation. And then there are some things in the textbook that we don't need to know. All right? And it also says in the PowerPoint. Okay, so this is the first little one that I found. Oh, my sound. Is it now because... That sound is up. Where else is sound? Remember, some of mine aren't downloading properly. They don't download properly. Okay, I'll do the narration. All right, so there we've got DNA replication occurring. All right, let's just go back a bit. When they gave me a new something here, some of my videos that I've uploaded last year and saved don't work this year. Don't ask me why. So I have to go and refine them. So obviously this is one of those. Okay, so here's DNA replication, and this enzyme is bringing those nucleotides in. Now, here they're saying base substitution. What happens if the wrong nucleotide was brought in? So instead of it being adenine with thymine, maybe guanine was brought in. Now, often there are enzymes that fix those kinds of mistakes up. Lots of enzymes fixing up the DNA, but what if that base was not fixed up? Okay, let's just see. So there's the one, okay, yes. All right, so this is called a point mutation, and they can happen, but often most times your cells fix them up. So that's what the DNA strand would have coded for. Remember protein synthesis? Serine, histidine, serine, valine. But now, when the DNA was copied, like when it's copied to make egg cells, or sperm cells, or when it's copied to make skin cells or any other somatic cell in your body, maybe the wrong nucleotide is brought in. So what is it going to affect? It's going to affect the, okay, now you've got to think back to protein synthesis. So the the DNA molecule opens up and it's copied by mRNA, remember? And then the mRNA goes to the ribosomes, and then at the ribosome is translated into a sequence of amino acids that are brought in by the transfer RNAs. You remember that? So they missed the middle bit out, and they've said, if this is the DNA molecule, and you understood that it was copied wrong just now. So there's one nucleotide here that is wrong, and the enzyme did not fix it. Okay, still taping. All right, so now it was serine, histidine, serine, valine. So if there's one base wrong, it's called a point mutation. Okay, now watch here. So instead, it's bringing in glutamine amino acid. Now that could change the protein, yes? But do you remember page number 17? Let's just go look there now. This is not how I was going to do it at all. This is just now happening. Okay, so on page 17, you've got that mRNA table. All right, so if you look there, where the genetic code is, there's many different codons coding for the same amino acid. So I'm going to write some of them here. So where's his? So C, that's not going to work. Oh, I took my pens down to the 10th. Hang on. In my bag. Okay. All right, so, So histidine, remember it was histidine here. Okay, sorry?
Page 17, right?
Hmm?
It's page
17, that table, yes. I'm looking there now. So go and find where it says hist, H-I-S. Can you see that there's two codons that code for the amino acid histidine? Can you see that? All right, so whatever this one was, so maybe it was that's the mRNA. Okay, I'm going to get myself, it's going to be too twisted here. So basically they're saying that nucleotide was right, that nucleotide was right, that one was brought in wrong. All right, so the mRNA will be wrong, so therefore the wrong amino acid is brought in. Okay? But now there are two codons that code for the same amino acid. So what if it was changed on the DNA to this one instead? It's still coding for histidine. Can you see how it might not be harmful then? But if you go to university, so you said you might do genetics.
That's Amy.
Oh, it's Amy, yes, sorry, you medicine. Yeah, don't worry, I didn't know that. Okay, so if you go, they're going to tell you about something called the Wobble Hypothesis, where even if it's a slightly different codon, the codon is slightly different, but it codes for the same amino acid. It's how it bonds the mRNA to the, how the tRNA is brought in, that it actually doesn't work. So that even having a different codon coding for the same amino acid, that resulting protein still doesn't work. But that's called the Wobble Hypothesis, and we don't do that yet. Okay, I only really recently, like 2 years, learned about that, or read about it. All right, so if the DNA was copied, and on the DNA it wouldn't be CAU, on the DNA it would be GTA. Do you remember this? GTG, you remember that? So there's the DNA, and it gets copied into messenger RNA, and then transfer RNAs bring the amino acids in. Okay, so So if the DNA was GTA originally, but instead when it was copied by replication, G was brought in, it will still code for histidine. That's okay, unless you learn about the Wobble Hypothesis. All right, but what they're showing here is that the wrong nucleic acid was brought in. So now it codes for a different amino acid, glutamine, not histidine. Okay, we got that. All right. Let's just see what else they do, maybe at the end. I'll download it again. Oh, so that first one was a substitution where the wrong nucleotide is brought in. That's called a substitution point mutation. Now watch this. See, I can't remember exactly the sequence, so let's go back a little bit. All right, so there's the DNA molecule. So when it gets copied, replicated before the cell divides, what if another nucleotide was put in? Can you see what it's done now? So we call this a frame shift. It's all in your textbook, don't worry. Can you see now that codon coded for serine, that one for glutamine, but this one is new, so all the rest afterwards are shifted. So every single amino acid after this is going to be different. That's a huge impact on the protein. Hi. Okay.
Just one.
That's fine. Did you escape? Okay. Yes. Okay, I'll go back as well. And it's going to be on the little PowerPoint that I'm going to show you as well. Okay, so there's the original DNA. So pretend that it was opened up and copied, and it was copied wrong. So the strand that you end up at the end with is wrong. Okay, watch it now. So when it was copied, another nucleotide was put in. Okay, so this one, that codon still will code for serine. That one will code for glutamine. You don't need to know the names, I just remember them. There's that new nucleotide that was put in by mistake. So could you see now that instead of this being the next codon and that one being the next codon, it's shifted?
So there's basically like an extra nucleotide.
Yeah. So that was the initial codon, this was the initial one, but now because that one has been added, it's shifted. So every single amino acid after that input of the wrong one, an extra one, is now different. That's called a frameshift mutation. It's an insertion of a nucleotide. The one just now, where one was swapped for another, is called a substitution.
How does that happen? How does an extra one just pop up?
Because our DNA molecules are opening up. Enzymes are bringing in the correct nucleotides, and sometimes they make mistakes. All right, and usually other enzymes in your cells correct those mistakes. Sometimes it doesn't. And then you end up with mutations, which could be, like you said, useful or whatever. What did you say? Useful or useful, beneficial, neutral, no change, like our color I still can see, or harmful, like causing cystic fibrosis or sickle cell anemia or something like that. Okay, well, sickle cell anemia is actually caused by parasites. All right. So let's just see what it does now. So every single amino acid thereafter is wrong. Do you want to see it again? Okay, oh wait, I missed that now. Or did it just go back? Let's just see what it does. So there, every single, in yellow, amino acid was different after that insertion of another nucleotide. So we call it frame shift.
Just pause now.
Oh, sorry. And there they've taken one away. Back to, I think it's back to original, hey? Yes. Oh, but now, what if when the DNA is copied, one is taken out? Could you see there was another frame shift there? Okay, let's just go back a little bit.
So is that the same as the frame shift?
It's a slightly different one. Watch it now. Right at the end. I can't. Okay, let's go there.
I think the video is too short to go back.
Yes, me too. Okay, there it goes. Okay, so now one was taken out. So now we've got that frame shift. Oh, that one, it was added in. Now one was taken out, and then you've got different amino acids afterwards. Okay. So they've got different names. I'll find the one with sound. I'll have to download it again, and I'll put it on. I'll put the link to it on Google Classroom. So I wonder if this other one can have sound. Maybe some of them, like once in a while. do I get problems with the videos I've saved already? I haven't had one like this since 2.1. It doesn't give me. So that's what we're talking about. Point mutation means it's one nucleotide. Wrong one put in, substitution. An extra one put in, insertion. And your textbook says addition. And the examiners, they say it's not maths, it's insertion. So the textbook is wrong. Okay, but I'll show you just now. And then deletion, a nucleotide taken out. So substitution, putting an extra nucleotide in or taking one out causes that frameshift. All the amino acids afterwards are wrong. But a substitution, like what I had on the board, might cause the wrong amino acid to be put in, or still the same one, because there's quite a few codons that sometimes code for the same amino acid. Okay, all right. 5 minutes.
The errors or mutations that occur in the replication of DNA can have significant impacts on individuals and on species. Mutations that occur within body cells can, for example, cause illnesses such as cancer if the mutation programs the cell and all its daughter cells to grow and divide uncontrollably. Such mutations are often caused in humans by radiation and chemicals such as those found in cigarette smoke that break strands of DNA. As we will see, the resulting repair work often leaves the DNA with an error. Mutations that occur in sex cells don't affect the individual in which they occur, but rather, if they are carried in a sex cell that unites with another sex cell, they are carried into the next generation. Such mutations have the potential not only to affect the individual that initially inherit them, but in the long term, the species as a whole. That is because an individual who inherits a mutation has a copy of that mutation in every cell of their body, including the ones they will eventually use in the production of sex cells. If the mutation increases the fitness, as judged by natural selection, of the individual and succeeding generations, the mutation will likely become more and more common in the species population. Each and every one of us are largely the result of mutations that, starting with the first piece of DNA in the first living organism, have been successful in increasing the fitness of individuals and species as judged by natural selection. We are, therefore, the products of successful mutations and the adaptations they led to. But history is full of even larger numbers of mutations that were judged by natural selection as unfit for survival. There are 3 basic types of mutations: point mutations, insertions, and deletions. Occasionally, during replication, in spite of all the proofreading and corrections, a mismatched base pair in one of the chromosomes, as for instance an adenine base pairing with a cytosine base, goes unnoticed, and the cell proceeds with either mitosis or meiosis. Repair enzymes in the daughter cell that received the mismatched pair notice the mismatch and proceed to correct it. However, these enzymes no longer have any way of knowing which strand is an original parental strand or a newly replicated daughter strand.
Okay, did anyone figure that earlier? So the enzymes come along and they try to fix it, but they don't know if this was the original DNA strand that was copied into that, or the other way around. So this you don't have to know. I just think it's interesting, okay? So they think they're fixing it, and then maybe they don't.
If the enzymes correct the mismatch by replacing the base on the daughter strand, the original base pair will be restored. However, if the enzymes correct the mismatch by replacing the base on the original strand, a base pair different from the original DNA will result, and a point mutation will have occurred. Insertions and deletions occur not due to any errors in replication, but rather from a break or breaks in one of the strands. Often outside sources, such as radiation, chemicals, cause these breaks. Insertions often occur when one strand breaks in a single location and one or both of the free ends loop out. Repair enzymes fill in the resulting gap with new nucleotides, making the strand that had the break longer than the strand that remained intact.
That seems like something out of something without fixing it properly, okay? Yeah. Okay, but you don't need to know this. We keep it simple. Swapping, inserting, taking out. We don't talk about how it loops and puts in the wrong ones in there.
As a result of how the process of replication works, after cellular division, one daughter cell will receive a new longer segment of DNA due to the insertion mutation, while the other will receive an unchanged, mutation-free segment of DNA. Deletion mutations occur if a strand of DNA breaks in two places. The section between the breaks is forced out, and the repair enzymes draw the two remaining strands together without filling in the missing nucleotides. Again, a loop occurs in one of the strands, but in this case, it is the intact strand. Now when cellular division occurs, one daughter will receive a shorter segment of DNA due to the deletion mutation, while the other strand remains unchanged. Having seen how DNA replicates itself and how mutations occur, we now look at how the genetic information contained in an individual's DNA is converted to proteins that make living organisms what they are.
Okay, so I just like that one because it showed a little bit better how it happens, but we don't need to know that. All right, go to page number in your textbook. There's a lovely introduction there. Have you got your books here? Okay, so where is it now? On page... where are we now? Oh, this thing. Sorry, my hands are freezing and I just, oh yeah, okay. It's on page number 124. I also scanned it yesterday, the book, and I put it on Google Classroom. So there's a lovely introduction on page 124. Do you need some more light? Okay. So types of mutations. When something goes wrong during the process of DNA replication, page 124. And it may result in a mutation. A mutation is a permanent change in the DNA of an organism. So it's not always a bad thing. It could be a good thing, like having those walruses or whatever they were, seals or whatever. They're stronger now, they can fight better, they can pass on those genes to their offspring, okay? And dominate more females and have more babies and things. All right, so it's a permanent change to the DNA. An average person, I've read 6. Okay, I forget here that it says 100. All right, but we're talking about little mutations. I think 6 big ones, okay? Then you get what we call somatic mutations and germline mutations. Somatic are the body cells, like your skin cells. So if you get sunlight radiation or X-rays or if you use sunbeds, that could damage the DNA and break it and form those loops. But germline mutation, the one little video showed that if the DNA in the egg or the sperm is mutated, then the new zygote and the new individual will pass that on, but it wasn't in the original mom or dad. All right. And some mutations occur spontaneously, DNA replications. Some of them have to happen because of mutagens, like smoking, all right, and chemicals and sunlight, okay? So those are mutagens, all right? And then it gives you some examples of radiation and chemicals. So X-rays, ultraviolet light, radioactive rays, chemicals, mustard gas. You've heard of mustard gas. Cause those horrible deformities. Pesticides, tar, DDT, and what is that name of the medicine that, no, hang on. Does that fit in here? The one that women took when my mom was pregnant, before that, they took a medicine to stop morning sickness and caused deformed children. Mutated the DNA. Thalidomide, I think it's called. All right. Think of people working at petrol stations, those fumes all the time. That's not good. That could cause, cells to mutate and become cancerous. All right, so you get small-scale mutations, and that's what we've kind of dealt with today. Point mutations, and then large-scale ones. And the large-scale ones I'll show you on Monday. We don't really need to know those ones. We've just done the swapping, the insertion, and the deletion. That's all we've done today. Okay, so we'll have a look on Monday. Are you nearly finished with your A3 summary? Okay, so do you need more time in class or do you want to do that at home? What do you want? Do it at home. So when should we go over it? New timetable. I'll see you for a triple on Monday. On Monday? Okay. And I don't see you on Tuesday, and then I see you for a single on Wednesday, and a triple, oh, it hasn't really changed, and a triple on Thursday, and a single, same place today. But this time it's not covered over by ASP, I can actually read it. All right, guys.
Thank you.
Okay.
Have a good weekend.
Yes, you too.